new brain discovery could help reverse the effects of aging. The promise of restoring youthful cognitive function has long driven neuroscience. Now, researchers at the National University of Singapore report identifying a microscopic regulator that may influence how well aging brains repair themselves. Their focus is a protein called DMTF1, which sits upstream of gene activity that enables neural stem cells to renew. These stem cells generate new neurons essential for learning and memory. With age, renewal slows, the stem cell pool becomes less active, and cognitive decline begins to take hold. The team found that restoring DMTF1 levels in older stem cells made them behave more like younger ones in laboratory tests. In the study, published in the peer-reviewed journal Science Advances, researchers examined human neural stem cells and lab models designed to mimic accelerated aging. They concentrated on telomere dysfunction, damage at chromosome ends widely considered a marker of biological aging. The findings were direct. Aged stem cells showed significantly lower DMTF1 levels. When DMTF1 was increased, the cells regained regenerative capacity. Further analysis revealed that DMTF1 likely works through helper genes ARID2 and SS18, which influence how tightly DNA is packed. Looser DNA packing allows growth and renewal genes to reactivate. When this pathway weakens, stem cells struggle to reset and multiply. Led by Assistant Professor Ong Sek Tong Derek, with Dr. Liang Yajing as first author, the research was conducted at NUS Medicine's Department of Physiology and the Healthy Longevity Translational Research Program. The findings remain largely cell-based, and the team stops short of claiming brain aging has been reversed. Next steps include testing whether boosting DMTF1 improves neural stem cell numbers, learning, and memory in aging models, while carefully monitoring tumor risk. Long-term, researchers aim to develop small molecules that safely stimulate DMTF1 activity for therapeutic use.